A person diagnosed with Alzheimer's disease in their 80s carries nearly the exact same functional genetic sequence they did in their 20s. Somatic mutations do accumulate in the human brain over a lifetime, but scattered text deletions cannot explain a highly stereotyped, region-by-region wave of neurodegeneration that unfolds over 30 years. The genome's text is not mutating. Instead, the cell is forgetting how to read it. Alzheimer's is a disease of the epigenome, a progressive corruption of the annotated score that tells neurons and glia which of their 20,000 genes to express and which to keep silent. This diagram illustrates the relationship between the two layers. The digital information, the DNA double helix itself, remains perfectly intact, but layered on top of it is analog information, a fragile pattern of methyl groups and acetylated histone tails. Unlike the DNA sequence, this epigenetic layer has no error-correcting backup. When these marks degrade, the instructions themselves become noisy. This split view correlates traditional pathology with the underlying data layer. While amyloid accumulates on the left, the right side tracks the corresponding collapse of enhancer landscapes and chromatin accessibility. The disease is defined by this drift into informational noise. This shift in perspective is significant. Because the genetic sequence remains unbroken, the pathology resides in how that sequence is read. In theory, a corrupted score can be corrected in ways a missing or mutated gene cannot. Reading the epigenome in postmortem human brain tissue introduces two major methodological hazards. The first is a cell composition artifact. When neurons die and microglia multiply, bulk tissue analysis registers a massive shift in methylation, which is really just a change in the local cell census. The second hazard is reverse causation. Tissue at autopsy is end-stage, making it difficult to distinguish an epigenetic mark that drove the disease from a mark that shifted simply because the neuron was already dying. To separate the signal from the noise, this matrix sorts the field's discoveries into a strict validity ledger. Bulk methylation hits, heavily exposed to these two confounds, fall into the correlational tier. Conversely, mechanisms like the HDAC2 blockade and transposon activation, which have been proven via interventional rescue experiments in animal models, sort into the strong causal tier. To map the true temporal architecture of the disease, we will follow these causal phase-mapped mechanisms. We have to strip away the biochemical noise of dying cells to see how the score actually fades. The biological origin point of Alzheimer's occurs decades before clinical symptoms appear, located deep in the brainstem. The highly arborized amineric neurons of the locus ceruleus operate under immense metabolic strain, making them the first cells to experience bioenergetic failure. That failure matters because epigenetic maintenance runs entirely on metabolic cofactors. Sirtuin deacetylases guard the genome, but they do not work for free. To strip acetyl groups from histone proteins, a sirtuin must stoichiometrically consume a molecule of NAD+. This gauge tracks NAD+ and S-adenosylmethionine. When these metabolic pools deplete, sirtuins abruptly halt. Without their activity, tightly wound heterochromatin unspools. As this metabolic ink runs dry, an isolated acceleration curve of epigenetic drift spikes within this specific brainstem nucleus. The bioenergetic collapse of the locus ceruleus is inextricably an epigenetic ignition point. The neuron hasn't lost its structural integrity yet. It has lost the molecular currency required to keep its dark genome silent. From the brainstem, the locus ceruleus projects upward, transsynaptically seeding misfolded tau proteins directly into the forebrain. Bess Frost's laboratory uncovered a mechanical consequence of this spread. Pathogenic tau overstabilizes the F-actin cytoskeleton, forcing the normally flexible cellular cytoplasm into a rigid, locked state. That rigidity creates a mechanical crisis. Through the link complex, the stiffened cytoplasm transmits intense mechanical force directly into the nucleus, physically invaginating the nuclear envelope and pulling it inward. This physical distortion depletes lamin B1, the structural scaffolding of the inner nucleus. Because constitutive heterochromatin is tethered to the lamina, this envelope collapse literally tears the dense chromatin open. The transsynaptic spread of tau into the hippocampus delivers a coordinated two-part payload. It provides a misfolding template, and it exerts a direct chromatin-loosening force on the vulnerable recipient neuron. As pathology reaches the hippocampus, local microglia abandon their neuroprotective homeostatic state. This shift is driven by an epigenetic reprogramming event, where active enhancer landscapes defining microglial identity collapse, biasing cells toward a chronically diseased state. This diagram visualizes the consequence of heterochromatin loss. With chromatin loose, ancient retrotransposons like line 1 and HERV-K in the dark genome are de-repressed, waking up to rapidly transcribe themselves. Acting like molecular parasites, these elements reverse transcribe their RNA back into DNA. The cytoplasm quickly floods with rogue cDNA and double-stranded RNA. The cell recognizes these nucleic acids using its innate immune sensors. Tricked into sensing an active viral infection, the neuron triggers a massive outward burst of type 1 interferon. This phenomenon is called viral mimicry. It is the specific engine behind the senescence-associated secretory phenotype, saturating the local microenvironment in an inflammatory bath. The epigenetic drift forces the neuron to sound an antiviral alarm against its own unlocked genome, directly causing the neuroinflammatory cascade that defines the middle stages of the disease. The terminal stage of Alzheimer's unfolds in the cortical networks. Historically, this phase is characterized by the structural disintegration of the synapse, leading to deep cognitive decline. But at the genomic level, this synaptic loss is an active event. It is an epigenetic clamping, the aggressive repression of specific gene programs required for synaptic plasticity. This diagram outlines Li Hui Tsai and Johannes Graf's discovery of the epigenetic blockade of cognition. We see an elevated concentration of the enzyme HDAC2 settling onto plasticity promoters. It strips away their activating acetylation marks, locking the structural spine genes securely shut. As crucial plasticity genes clamp shut, the neuron's core identity dissolves. Off-target cell cycle genes inappropriately relax and activate, forcing a doomed attempt to replicate that triggers terminal cell death. The definitive proof that this is a disease of misread information comes from intervention. When researchers pharmacologically block or knock down HDAC2, plasticity genes reopen, synapses regrow, and memory function is rescued. Terminal dementia behaves as a sharp threshold because the disease eventually enters a self-reinforcing epigenetic lock that cannot be undone. The cell has rewritten its own operational score and now plays the pathology strictly from memory. From the early metabolic strain in the brainstem to the terminal silencing of the cortical synapse, Alzheimer's operates as a single continuous system of analog information loss. This timeline translates that biology into a phase-matched clinical roadmap. Rather than attempting to clear a downstream protein, the goal is to rewrite the epigenetic score based on the specific decade of progression. Early interventions focus on restoring NAD+ and methyl donors to maintain the metabolic ink. Mid-stage therapies target TGF-beta signaling to reverse microglial reprogramming. In the late stages, the intervention relies on physically blocking the viral mimicry. This is the biological rationale for current clinical trials, repurposing HIV nucleoside reverse transcriptase inhibitors, like lamivudine, to stop the line 1 transcription process and cut off the inflammatory signaling at its source. Because the core damage consists of miswritten instructions, a phase-matched rewriting of those instructions is the only logical clinical response. A radical extension of the information theory involves partial epigenetic reprogramming. This relies on the discovery by Shinya Yamanaka, seen here holding his Nobel Prize, that mature cells can be returned to a more youthful state using specific transcription factors. Translating this to the human brain carries severe theoretical risks. Push the reprogramming too far, and the neuron completely forgets its post-mitotic identity, creating an extreme risk for oncogenesis and teratomas. Alzheimer's is a disease of corrupted pattern. The instructions for maintaining a healthy neural network have degraded into chemical static, but the baseline sequence beneath that static remains intact. The instructions are not deleted, merely unreadable. Because the pattern still exists, the lost architecture of the mind is the one thing in neurodegeneration that can, in principle, be written back.